Hi students, in this chapter, chapter 17, we're going to be talking about the reproductive system, and these are the topics that we're going to be discussing. We'll talk about gametogenesis, which is the process of making gametes, whether a sperm or an egg, sex determination and sex differentiation. So how does a fetus or in utero, how do the genes determine what gender the baby is going to be? And then we'll talk about the physiology of the male reproductive system, followed by the physiology of the female reproductive system. So at this point, if you're a student that did not, or that needs a refresher on the anatomy of the reproductive system, I would definitely recommend that you do so before listening to this lecture. So first topic is an overview of gametogenesis. So all of our cells, have 46 chromosomes. In order to make gametes, in order to make gonadal cells, which again would be a sperm or an egg, we have to split those chromosomes in half so that each sperm or each egg would have only 23 chromosomes, so that if fertilization occurs and these chromosomes are all added up together, we go back to those 46 chromosomes, which is the genetic makeup of all of our cells. Okay, so in order to do so, we have to go through what is known as a meiotic division. Meiotic division is a division where we split the chromosomes in half. It occurs, it's a process that has a first meiotic division and then there's a second meiotic division. So your first meiotic division would be where you're splitting these chromosomes in half. So 46 chromosomes, you're going to end up at the end of their first meiotic division having two cells with 23 chromosomes each. But the thing is that if you look at the chromosomes, each chromosome looks like an X. So each chromosome is made out of two chromatids that are attached in the middle by a centromere. And that's not what we want. We want cells that look like these that have 23 chromosomes, but each chromosome is just made out of one thread, made out of one chromatid. And that is why we have to go through this second stage of meiotic division, and that is, again, to split those chromatids in half. So at the end of your first meiotic division, you get two cells. Each cell has 23 chromosomes, two chromatids. But at the end of your second meiotic division, you have four cells. Each cell has 23 chromosomes, one chromatid each. Okay? In the case of spermatogenesis, these spermatids, which are, baby or kid sperms, they would have to mature into a sperm cell, and that process is known as spermiogenesis. And we'll get to more details about each of these processes when we kind of get to that part. In the female system, we end up making just one egg, okay? We do not make four cells. You see here that we're ending up making just one egg and three polar bodies. These polar bodies are a way to remove all of that extra genetic material. Okay, so they have no function. They are going to die off and be absorbed by the tissues, be absorbed by the body. But again, we have to remove all of that extra genetic material in order to get that egg that has your 23 chromosomes with one chromatid each. Now, in order for us to not be carbon copies of our parents, or our offspring to be carbon copies of us, we need to have there has to be a way where we can mix up the genes. And that mixing up of genes is known as crossing over. That occurs during the first meiotic division. And if you want to be specific about it, it occurs during prophase 1. So if you guys remember the mitotic division, there was prophase, metaphase, anaphase, and telophase. And to remember that, there are several mnemonics, but I prefer, I passed my anatomy test. So I stood for interphase, P was prophase, M was metaphase, and then anaphase and telophase. Meiotic division, again, as we said, we have first and second stage, and first stage would go through pro, meta, ana, and telophase. So would your second meiotic division, so we call them prophase 1 or prophase 2, metaphase 1 or metaphase 2, and so on. So crossing over occurs during prophase 1. And again, this is where pretty much the X chromosomes, they overlap, and there is a little bit of mixing up of the genetic material together. They share and they they share the genetic material in order, again, to make these gametes that have a good mixture so that the offspring there's a variation among the offspring. And not only does crossing over happen in spermatogenesis, it also happens in oogenesis as well. Just a quick tip, in oogenesis, and we'll get to that more detail when we reach that part, it goes through the first meiotic division and then it stops. Okay, second meiotic division does not occur in oogenesis unless fertilization is going to happen, and that's why you see that sperm cell in that picture. Although we're talking about making eggs. In that case, if fertilization is going to happen, this secondary oocyte is going to go through a secondary meiotic division really fast in order to split again those chromatids, and there's going to be a fusion between the genetic material in the sperm and the genetic material of the egg in order to make that zygote that is now 46 chromosomes. So let's talk a little bit about sex determination, meaning how is the sex of the fetus determined? So the sex chromosomes are X and Y. Okay, in males there is one X and one Y chromosome, and in females there are two X chromosomes. In utero at the age of 7 weeks, okay, if the Y chromosome is present, there is a gene known as the SRY gene. SRY stands for the sex-determining region of the Y gene. That is going to make a couple of hormones so that that fetus is going to develop into a male fetus. In the absence of the Y chromosome, in the absence of this SRY gene, the fetus is going to develop into a female. So I like to think about it that by default, all fetuses are going to develop into females unless there is this SRY gene that is found on your Y gene, on your Y chromosome. Okay. So here's a summary of the differentiation. I'm actually going to go to the picture and then go back to that one slide. So this is the picture of a fetus at the age of 5 to 6 weeks. Again, you cannot tell the gender or you can't tell the sex of this baby by looking at any of the structures. What they actually have, if you look at this blue and orange, these are known as gonadal ridges. These gonadal ridges have the ability to either develop into an into an ovary or into a testis. There's also this lighter blue known as the Wolffian duct, and there is this purple known as a Mullerian duct. So the fetus has all of the material to develop into either a male or a female. There's also the urogenital duct. This part is going to be develop into the urinary system. So we'll see how the development of the urinary system is very closely related to the development of the reproductive system. Okay, so what happens if their genetic makeup is an XX, meaning that this is going to end up becoming a female? So what happens is that the gonadal ridges, so again these blue and that orange, are going to develop to make the ovaries, and then the Mullerian ducts develop into the rest of the female system. So you can see here the fimbria, the fallopian tube. This is going to eventually develop into the uterus, as you can see down here, and then the cervix and the upper part of the vagina. But you will see here that there is the disintegration or the deterioration of the Wolffian duct. The part of the urinary system that we talked about, this is going to end up making the urinary bladder, and it will also kind of deflect back in order to make the lower part of the vagina. In the male system, in the presence of the Y gene, okay, there is SRY gene, and that is, like I said, is going to make a couple of hormones, and those hormones are known as your AMH, anti-Müllerian hormone, okay, which is going to, from the name, it's going to lead to the deterioration or the regression of the Müllerian duct. Remember, the Müllerian duct made parts of the female system, so we're not going to need that because this is going to be a male. So the AMH is going to lead to the regression of the Müllerian duct, and then it's going to also lead to the production of testosterone by the gonadal ridges, and that's going to lead to the differentiation and the growth of the Wolffian duct into the different parts of the male system. And you can see that here, the gonadal ridge is going to make the testis, and the testis is going to descend down into the scrotum, while you can see here the deterioration of the Müllerian duct, while the Wolffian duct has developed into the different parts. So you could see here your epididymis, the vas deferens, the seminal vesicles, and so on. While the part of the urinary system, the urogenital ridge made the urinary bladder, and again, obviously the urethra, where all of this, where the male system is going to connect in order to dump the semen later on. So going back to this, okay, in the presence of an XX chromosome, which basically means there is no SRY gene, that fetus is going to develop, the primordial gonads are going to develop into the, I want to find the ovary here on this, but I can't find it, but the primordial gonads are going to develop into the ovaries. And since we do not have testosterone and there is no AMH, the Wolffian duct is going to regress, while the Müllerian duct is going to transform into the different parts of the female system. While your urogenital ridge is going to make the outer part of the female reproductive system and the lower part of the vagina. In an XY chromosome fetus, that SRY gene is going to differentiate the gonadal ridge into a testis, and those cells are going to start making AMH, which is going to regress the Müllerian duct. And then it's also going to make testosterone, and that testosterone is going to stimulate the Wolffian duct to transform into the parts of the male system. So what are some disorders of sexual differentiation? So sexual differentiation means that there does not have to be there could be a difference between the genotype and the phenotype of the baby. So a genotype means what does the gene tell? What do the genes tell me? A genotype would be just looking at XX or XY. Okay, there are other variations of that, but we're not going to get into it. So if the genotype is an XY, that would be the genetic, that would be what the genes are showing me. Phenotype means what does that baby look like from the outside? Okay, so phenotype. So genetics are more chromosomal, while the phenotype means like the structure on the outside. So we've got these individuals that have what is known as androgen insensitive syndrome. Okay, so genotypically they are males. Okay, the testis secretes androgen, but there is a mutation of the genes where the androgen receptors are not responding to that androgen. So no matter how much androgen is present, the receptors are not responding to that. So in these babies, they would have what is known as an undescended testis, and they remember that testosterone is what stimulated the Wolffian duct to transform into the different parts of the male system. And it also helped in the development of the penis and the scrotum and the prostate gland. So even though these individuals have testosterone, since the receptors are not responding to it, that means that these structures are not going to develop. So phenotypically, looking at the baby from the outside, they are phenotypically females. So that is one abnormality we want to talk about. Another disorder is known as congenital adrenal hyperplasia. This is the opposite, where genotypically looking at the chromosomes, XX, so they're females, but there's a gene mutation in the cortisol pathway. So I kind of borrowed this from the endocrine system so that you could see here that testosterone, in order to make estrogen, okay, we have to make testosterone first. And then there is this enzyme known as aromatase enzyme that will convert testosterone into estradiol, or which is a kind of estrogen. Okay, these, in this congenital adrenal hyperplasia, there's a gene mutation in the cortisol pathway. So, since we have that same precursor, okay, we're going to kind of go down towards making testosterone. So there is a mutation in this step right here. It's the step where we're trying to transform the testosterone into estradiol, and we're unable to do so. So that is going to lead to the accumulation of testosterone. So these individuals are going to be genotypically females, but phenotypically they would be males or ambiguous, meaning that you can't really tell from the outside if they have female external genitalia or male external genitalia. So what are the stages in the control of the reproductive function? There is in utero, okay, gonadotropin releasing hormones and gonadotropins are secreted at relatively high levels, okay, especially in XY fetuses where those testosterone is going to lead to most of the development. From childhood to the onset of puberty, those levels are relatively low. Okay, it's not until puberty hits, and that's when the gonadotropin releasing hormone from the hypothalamus is going to kind of trigger. That's your on switch for triggering puberty, and this is the period of time of fertility. As we age, the reproductive system tends to slow down, okay, especially in females where there is entire cessation of oogenesis, and that is known as menopause. There is in some men, there is kind of like an equivalent of menopause that's known as andropause. It is not common, but there is, as males tend to age, there is also that decline, slight decline in the spermatogenesis area. Some even kind of stop that totally, but that is something that is not as common as menopause. Menopause is something that every woman experiences. Andropause is something that only some men experience. So here's the anatomy of the male reproductive system. Just to go through it really quick, there's the testis, the epididymis, the vas deferens or the ductus deferens that passes above the urinary bladder all the way to the back. And then together with the seminal vesicles, all of those are going to be dumped into the ejaculatory duct through the prostate gland, and then through the urethra, and the semen will pass through the penis all the way to the external urethral orifice. Now back to the summary of spermatogenesis. So remember I said I'm going to kind of talk about it individually. So we start off with these very primitive cells, and those are known as spermatogonia. Those have 46 chromosomes, 2 chromatids each, and we have to split those to end up making cells that only have 23 chromosomes with 1 chromatid. So that spermatogonia, through mitosis, is going to make primary spermatocyte. Remember, mitosis does not involve any splitting of chromosomes, okay? We're just making copies of these spermatogonia. They grow up a little bit, though. They differentiate, so we're going to call them primary spermatocytes. And then these primary spermatocytes are the cells that do undergo your first meiotic division to make these secondary spermatocytes. And you see here there's a split in the number of chromosomes, but the number of chromatids are still 2. And that's why we have to do the second meiotic division to make these spermatids, which I tend to remember as the kid sperms. So now they have the 23 chromosomes with 1 chromatid, and then that spermatid has to differentiate into a mature sperm that has the head, the the neck or the midpiece, and the tail. Okay, this process of maturation from a spermatid to spermatozoa is known as spermiogenesis. Okay, so this word right here, spermiogenesis, while the rest is known as spermatogenesis. This is what a human mature sperm looks like. So there's the head and a midpiece and the tail. The tail is known as the flagellum, and it's there to help with the swimming process. If you take a closer look at the head, which we'll do right over here, so close up of the head of the sperm, that's where the genetic material is. So you'll find the nucleus here, the 23 chromosomes. Each chromosome is made out of only one chromatid. And on top, there's an acrosome. That acrosome has lots of enzymes that'll help in the fertilization process, help kind of penetrate that egg so that we can reach the egg and fertilize it. The midpiece has lots of mitochondria in order to supply the energy needed for the swimming in order to reach the egg. So how is it that the male reproductive system is controlled hormonal-wise? Well, everything, as I said, starts at the hypothalamus. So the hypothalamus makes gonadotropin-releasing hormone. That is going to reach the anterior pituitary, and it'll stimulate the anterior pituitary to make FSH and LH. So FSH, although it stands for follicle stimulating hormone, and LH stands for luteinizing hormone, and those terms tend to be to occur in the female, but they are the same hormones that are made in males. FSH is going to reach the testis and stimulate the Sertoli cells to undergo spermatogenesis, to help or stimulate the process of spermatogenesis. Okay, so it's easy to remember that all of these things that have an S, okay, so FSH, stimulate Sertoli cells to stimulate spermatogenesis. Now the Sertoli cell is also, not only does it stimulate spermatogenesis, it also makes inhibin, which is a hormone. Inhibin is a hormone that inhibits the anterior pituitary and inhibits it, the FSH production from it. It does not have an effect on luteinizing hormone. The anterior pituitary is also going to make LH. LH is going to stimulate the Leydig cells that are found in the testis to make testosterone. And then testosterone is going to, the target cell of testosterone would be the reproductive tracts and the other organs, and it will lead to the development and the maintenance of the male sexual characteristics. Testosterone, by negative feedback, will inhibit the anterior pituitary from making LH. It also inhibits the hypothalamus from making gonadotropin-releasing hormone. So you see here again, all this negative feedback happening. Now the functions of the Sertoli cells. So just to make sure that we understand what these Sertoli cells and Leydig cells are, the structure, and that's why again I asked you to go back and kind of take a look at the basic anatomy of the male system. In the testis, there are several cells. Some of them, we've got spermatogonia. Spermatogonia, those are the cells that are line the seminiferous tubules, and these are the ones that undergo spermatogenesis, excuse me, and end up making the sperm. So you can see the sperms with the tails kind of like more towards the middle. The spermatogonia are more towards the basement membrane of the seminiferous tubules. And then the testis also has the Sertoli cells. So these Sertoli cells are cells that support the spermatogonia, and they release inhibin, and we'll take a look at the other functions that Sertoli cells do. The interstitial cells of Leydig, or also just known as Leydig cells, those are the cells that release testosterone. Okay, so these are the three different cells that are available that we're going to be, that we're discussing. So here in this slide, we're going to talk about the function of Sertoli cells. In the fetus, these are the cells that make AMH. Okay, AMH has another name, which is Mullerian inhibiting substance, or MIS. Okay, and those, again, are responsible, these are the same hormone, okay, but though it is responsible for getting rid, or the degradation, the atrophy of the Mullerian duct, so that the Wolffian duct will respond to testosterone and develop into the male system. So what is the function of the Sertoli cells in adults? Well, in adults, like I said, it supports the process of spermatogonia. It is going to, and we'll see how that kind of happens. It makes inhibin. Inhibin is going to inhibit the making of FSH. Okay, it has no effect on LH secretion from the pituitary gland. It acts as a barrier to chemicals. So it's kind of like protective to the spermatogonia. It also has a phagocytic function, so it'll try to eat up, engulf any foreign bodies trying to reach the spermatogonia. It develops or it nourishes the sperms as well. Okay, it responds to testosterone and to FSH, so we already know that it responds to FSH, and it makes a paracrine chemical. So remember, paracrine is a hormone that affects neighboring cells. So, and that is how it stimulates spermatogonia to proliferate and differentiate and go through the meiotic division. Okay, it also secretes different paracrines to stimulate the Leydig cells, their neighboring Leydig cells, to produce testosterone. So what are the effects or the actions of testosterone in males? Okay, well, it's definitely needed for the process of spermatogenesis. By negative feedback, we said it already inhibits LH production in the anterior pituitary, and it also inhibits the production of gonadotropin, releasing hormone from the hypothalamus, which are the functions that we talked about here. So testosterone inhibiting LH and also inhibiting gonadotropin releasing hormone. It starts off, okay, the differentiation of the development and the maintenance of the male sexual characteristics. Okay, for example, the enlargement of the laryngeal prominence, which is Adam's apple, and the hoarseness of voice, the change of voice that occurs, the facial hair, for example. So all of these, the development of these sexual characteristics and the maintenance of it throughout life, that is the function of testosterone. It also increases protein anabolism. Okay, so making of muscle fiber, muscle protein, growth of bone, and then the cessation of bone growth. Okay, so it is going to lead to that growth spurt that happens. And then it leads to the complete ossification of the epiphyseal plates. And your epiphyseal plates, those are the growth plates that are found in bones. So what it does is that, first off, it rapidly increases the growth of bones, and then it stops it. Okay. Testosterone is also needed for sex drive, and very high levels of testosterone can lead to aggressive behavior. Testosterone also stimulates the stimulates the kidneys to make erythropoietin, and erythropoietin would stimulate the bone marrow to make more RBCs, and that's why males on average have a higher RBC count, because of the effect of testosterone on the stimulation of erythropoietin by the kidney. So other effects, in some tissue, testosterone has to be converted in order to be used. So some cells of the body do not really have testosterone receptors, but remember that we are able to change testosterone into estrogens by an enzyme known as aromatase enzyme. So if you guys remember that slide that we borrowed from chapter 11, if cells have aromatase enzyme, that would change testosterone into estrogen. And some cells of the body need that because they do not have testosterone receptors. For example, in the brain, okay, brains do not have testosterone receptors. They would have to be converted by that aromatase enzyme into estrogen. And that is going to, that is the, so to speak, kind of the active form of testosterone in brain cells. In the prostate, testosterone has to be activated into a more potent kind of hormone, and that is known as dihydrotestosterone. So you're basically going to add 2 hydroxyl groups, and that enzyme is known as 5-alpha reductase. Okay, high levels of DHT, though, worsens male pattern baldness. Okay, and some prostate cancer cells are stimulated by DHT. So high levels of DHT can lead to male baldness, can also lead to aggressive pancreatic cancer. So that is why inhibitors of that enzyme, so inhibitors of this 5-alpha reductase enzyme, can be used in the treatment of male pattern baldness and also in the treatment of some kinds of prostate cancer. Now for puberty in males, well, puberty, as I said, whether male or female, really, it's because that on button, you have to turn the on button in the hypothalamus. So once the hypothalamus starts making gonadotropin releasing hormone, that is going to stimulate the anterior pituitary and so on, and the story continues. In males, on average, it occurs between the ages of 12 and 16. It's very variable, though. It varies by race and by ethnicity and, family history and so on. But on average, the range is usually between the ages of 12 to 16. We'll talk a little bit about the pathway for erection and then followed by the pathway for ejaculation. So for an erection to begin, there is either input from locally from the penis through mechanoreceptors, or it could be coming from the parasympathetic stimulation in the nervous system, where it could be, thoughts or emotions or sight or smells. Those impulses are going to stimulate the production of nitric oxide, and nitric oxide is a vasodilator. It'll lead to the dilation of the arteries, and that dilation will bring in blood to the area, and that's what leads to an erection. So erection is really under mainly the control of the parasympathetic stimulation. While ejaculation, though, you'll find that it is more under the control of the sympathetic nervous system. And ejaculation means pushing the sperms that have actually been stored in the epididymis. You'd have to push all of the sperms through the vas deferens, and then kind of go back to that picture to understand how that is. So when sperms mature in the epididymis, they remain there for a while, and then upon ejaculation, they would have to be pushed through the contractions of smooth muscles through the vas deferens, and it starts to collect different secretions from the seminal vesicles, from the prostate gland, and the two tiny glands that are found here known as the bulbourethral glands, and then through the urethra. So for all of that to occur, you need contractions, peristaltic waves of contraction of the smooth muscle fibers, and that's what ejaculation is. And that would be under the control of the sympathetic nervous system. So emission is that contraction that I was just talking about, the smooth muscles of the epididymis, vas deferens, until it is pushed out. And semen is made out of, the volume of it is about 3 milliliters, and it contains about 300 million sperms. And in order to prevent, kind of as a method of contraception, there is a procedure known as vasectomy, which is basically cutting the vas deferens. Okay, and do know, though, that it does not affect spermatogenesis. Okay, so I know I'm going back and forth on you guys, but if the vas deferens is cut, okay, that doesn't mean that the testis has been ruined. The testis is fine. It's still going to be making sperms. Those sperms, though, are going to be stored in the epididymis, and even during during ejaculation, though that's where they do not have the means to reach the outside of the body. Okay, so those are going to remain in there. They are going to die off after a while and then be absorbed by the body. So we talked about these. Now a little bit about the effective abuse of anabolic steroids in male athletics. So although from the outside, the use of steroids kind of looks like a good idea because it increases muscle mass, which obviously will increase athletic performance. But the thing is that if you take steroids from the outside, okay, if you take, an exogenous source of steroids, those steroids are going to be converted into testosterone, okay? And that means that gives your body the wrong signal to the testis that it does not need to make its own testosterone. So if this abuse remains for a long period of time, that can lead to shrinking of the size of the testis, so decrease in the testicular size, and then on the long run would lead to a low sperm count. Okay, now we're going to talk about the anatomy and physiology of the female system. Again, without going to the details of the anatomy, just the different parts. So there's the ovary, fallopian tube or the uterine tube, and then there's the uterus. The uterus is made out of the myometrium, and then there's the inner lining known as the endometrium, and that's the part that is shed every month during menstruation. There's the cervix, and that's the portion of the uterus that is projected into the upper part of the vagina. And then there's the vagina with the external vaginal orifice. The functions of the ovaries are oogenesis, which are the production of the eggs, okay, or the gametes. And that's where the oocytes mature. And then there's the ovulation, and that's the expulsion of the mature oocyte. And we'll kind of go into the details of what is the mature oocyte as opposed to the immature ones. And the ovaries also secrete the female sex hormones, estrogen and progesterone. Okay, and it also makes a protein hormone known as inhibin that we talked about in the male system. So you'll see that these hormones are pretty much the same. So the summary of oogenesis in the eggs. We start off with oogonia, okay, so just like in the spermatogenesis, we started off with spermatogonia. And the way I remember it is that these cells are going to make us eggs, okay, so oogonia are going to become eggs. So these oogonia are, again, 46 chromosomes, 2 chromatids. You have to make lots of copies of these, okay, so they undergo mitosis and differentiation into a primary oocyte. A primary oocyte still has the 46 chromosomes with 2 chromatids. And the process of oogenesis in females starts in utero, during fetal life. So you'll see here that these oogonia mature into primary oocytes, and female girls are born with primary oocytes started and halted or kind of frozen at that stage, okay? And it won't pick up until puberty starts. So again, the first steps, the oogonia becoming primary oocytes, those occur during fetal life. At birth, primary oocytes are stopped or kind of freeze at that stage, okay, where 46 chromosomes, 2 chromatids, and during childhood, those remain the same. It's not until puberty starts to kick in where the hypothalamus will make gonadotropin-releasing hormone, stimulating the production of FSH from the anterior pituitary. Now that is going to stimulate the remaining process of oogenesis. So you'll see here that it'll stimulate this primary oocyte to undergo the first meiotic division to make a secondary oocyte and the first polar body. Again, the polar body is without any function. The only function that it does is to remove the extra genetic material. And the secondary oocyte has 23 chromosomes with 2 chromatids each. Now this secondary oocyte is going to remain, this is what is ovulated, okay? You can see here that the secondary oocyte will not undergo the second meiotic division unless fertilization happens. So if fertilization does not occur, the secondary oocyte will not go through the second meiotic division. Okay, we're going to assume that fertilization occurred. Okay, so now the secondary oocyte will undergo the second meiotic division to make that ovum. The ovum has the 23 chromosomes with that 1 chromatid. This ovum is going to be fertilized by the sperm, and it will end up going back again to the 46 chromosomes. So again, in oogenesis, there are a couple of things to keep in mind. That the first process of going from oogonia to primary oocyte happens in utero. Okay, ovaries are born with the primary oocyte, and it remains as such during childhood. It's not until puberty hits where the primary oocyte will undergo the first meiotic division to make the secondary oocyte, and this is the stage that is ovulated. If fertilization does not occur, then this will die off and the second meiotic division will not happen. If fertilization happens, the secondary oocyte will undergo the second meiotic division to make that mature ovum. So at the end of oogenesis, we really end up making just one egg, while at the end of spermatogenesis, we ended up making 4 sperms. So this is looking at the development of the ovarian follicle and the oocyte. So know that the stages that we were talking about here, primary oocyte, secondary oocyte, ovum, so on, that only represents the blue part in the middle, okay? Everything else represents the maturation of this huge, what is called a mature follicle, okay? As what we are going to, what is ovulated again is this primary oocyte, okay? So, sorry, it was the secondary oocyte. So we have the oocyte here with these granulosa cells, and the granulosa cells are the cells that are involved in making estrogen. And you can see that that layer is going to thicken and surround, the outer surface is going to be surrounded what is known as theca cells, which are cells that start to make testosterone. And the mature follicle at the end has the secondary oocyte. It is surrounded by granulosa cells, and it has what is known as a zona pellucida, or a pale zone. It is surrounded on the outside by this by these granulosa cells that again make estrogen, and the theca cells that make testosterone. So what happens is that, so I don't want you guys to understand that the mature follicle makes testosterone. What happens is that the theca cells make testosterone. Testosterone diffuses into the granulosa cells, and by that enzyme aromatase, it'll change testosterone into estrogen. So what is eventually made by the egg is estrogen and not testosterone. Okay, if you take a look at this portion, and again, this is what is going to be eventually ovulated, and the rest of the mature follicle is going to stay in the ovary and become what is known as the corpus luteum, or the corpus luteum. We have the following layers, okay? So there would be some theca cells, and then granulosa cells, and then your zona pellucida, and then there would be the cell membrane of that oocyte. Now we're going to start looking at the summary of events that happen during the menstrual cycle. Again, this is only if fertilization does not occur. These are the names of the phases when it comes, taking into consideration what is going on in the ovary. These phases have different names in the uterus, so we'll take it one step at a time. We'll talk about the names of these phases in the ovary. Obviously, the main event that happens mid-cycle is ovulation, and we are going to, use an average of 28 days for a cycle. So at day 14, that's when ovulation is going to occur. Anything before day 14 is known as the follicular phase, which means that in the ovary, the follicle is starting to develop. Okay, that mature follicle that we just talked about is developing during this follicular phase. After ovulation occurs, remember we said that the remnants, or the remaining part of the follicle, is going to change into, or we'll now name it the corpus luteum, or the yellow body. And that's why we call this phase the luteal phase. So what really happens during this follicular phase? Well, first off, we have the first 5 to 7 days of that phase. That's where uterine bleeding happens, okay? That's when menstruation is occurring. So day 1 of the cycle is when bleeding starts. Then we have, again, on average, 5 to 7 days of menstruation. Okay, and this is also when the ovaries preparing for the next cycle. Okay, so some follicles, a good number of follicles are going to start the race to develop. So you'll see that multiple follicles are going to start to develop. About a week later, okay, so by the time that menses is over, only one follicle becomes the more dominant and the rest degenerate. Okay, and that follicle is going to mature and become that Graafian follicle that we spoke about right here. Okay, during this stage, that follicle is making estrogen. Okay, so this portion of the cycle, follicular phase, is mainly, the ovaries are mainly making estrogen. Yes, it's making a little bit of progesterone, but the main hormone coming out of the ovaries at this time is estrogen. Estrogen is going to be circulating throughout the whole body, and it will reach the uterus, the endometrial lining or the inner lining of the uterus, and it will lead to its proliferation. That is why it is known as the proliferative phase in the uterus. So the follicular phase in the ovary coincides with the proliferative phase in the uterus. Okay, now that ovulation happened, okay, we've ovulated the secondary oocyte. Now the remnants or the remaining part of that mature follicle is going to become the corpus luteum. It'll make mainly progesterone. So the luteal phase is mainly under the influence of progesterone. So you see here that the corpus luteum is going to function. It'll keep on functioning up until, the very end of that phase. And then the corpus luteum is going to degenerate. Again, keeping in consideration that pregnancy or fertilization did not occur. If fertilization did occur, the corpus luteum will keep on making progesterone for about 3 months until the placenta is fully developed and the placenta can take over that function of making progesterone to maintain the pregnancy. But again, this is keeping into consideration that fertilization didn't happen. So during this luteal phase, as we said, the corpus luteum is making mainly progesterone. So keep in the back of your head that it's also making some estrogen, but mainly progesterone. Now, progesterone circulates to the rest of the body, and it leads to the increase in the number of glands in the endometrium. And that is why that phase, or those changes in the uterus, are known as the secretory phase. So the luteal phase in the ovary corresponds to the secretory phase in the uterus. Okay, this graph shows us the different levels of hormones. Okay, so all the way down here, we are talking about the ovarian phases. There's the follicular phase, which is the first half, and then we have ovulation in the middle, and then there's the luteal phase. If you look at the different hormone levels coming out of the ovary, you'll see here that the follicular phase, it's mainly estrogen. Progesterone is there, but it's not a lot. Okay, and then towards the end of the follicular phase, now this is where that one Graafian follicle is starting to mature, and the rest just degenerated. You see here it reaches a peak right before ovulation. At the time of ovulation, after ovulation, estrogen levels tend to go down while progesterone levels go up. Okay, at the end of the luteal phase, both estrogen and progesterone go down. And that is why the endometrium is going to be shed. The endometrial lining of the uterus is proliferating and is growing because of the effect of estrogen and progesterone. When there is no longer estrogen and progesterone to maintain it, these cells are going to literally die and then shed off and lead to menstruation. So let's kind of, go into a scenario where these levels do not go down. Well, then menstruation will not occur. Okay, if the body is able to maintain these levels of estrogen and progesterone, and that's what occurs during pregnancy, where the corpus luteum keeps on making progesterone, keeping those levels up, and maintaining the endometrium in order for in order for implantation to occur. Now, this graph up here is showing you the levels of FSH and LH coming from the anterior pituitary. So you'll see here that FSH, the orange line, okay, is going up, and that is what's stimulating the maturation of the follicles. And then you'll find that LH is kind of constant until there's that spike or what is known as the LH surge right before ovulation, and that LH surge is what stimulates ovulation to happen. And then those hormones tend to go down gradually as well up until the end of the cycle. So how is it that there's a control of estrogen synthesis during early and middle follicular phases? So we're talking only about the follicular phase, okay? LH is going to stimulate theca cells to make androgen, which are the outer cells of that Graafian follicle or that ovarian follicle. And then the androgen is going to diffuse into the granulosa cells. And the granulosa cells are stimulated by FSH to convert androgen into estrogen. So this is what I kind of mentioned over here, where these theca cells are making testosterone under the influence of luteinizing hormone. That androgen is going to diffuse to these granulosa cells, and under the effect of FSH, these granulosa cells are going to convert androgen into estrogen. And that occurs by the action of aromatase enzyme. Now for the bigger picture of things, so what happens at puberty, you have the gonadotropin releasing hormone being released from the hypothalamus. It'll stimulate anterior pituitary to make FSH. FSH, well, let's do it the other way. We'll start at LH. LH is going to stimulate your theca cells to make androgens. Androgens diffuse to the granulosa cells. These cells are stimulated by FSH to make estrogen. Okay, and estrogen is responsible for all of the female sexual characteristics. And these granulosa cells also make inhibin. Inhibin is going to inhibit the production of FSH from the anterior pituitary, while estrogen is going to inhibit the production of FSH and LH and the production of gonadotropin releasing hormone from the hypothalamus. Now, how is it that this LH surge occurs? Excuse me. So the ovaries are making large amounts of estrogen, okay, and those large amounts of estrogen are going to stimulate the anterior pituitary through positive feedback to make more LH, and also stimulate the hypothalamus to make more gonadotropin releasing hormone. The push to make more LH is what causes that LH surge. Okay, that LH surge leads to the ovulation, and then again the rest, so we've ovulated the secondary oocyte, the rest of the mature follicle changes into this corpus luteum that is going to make progesterone and estrogen, but mainly progesterone, and now we are into the second part, which is the luteal phase. Now, how is it that the progesterone is going to inhibit the production of hormones so that this cycle can end? Well, that progesterone is, by negative feedback, is going to inhibit FSH and LH. It is also going to inhibit the gonadotropin releasing hormone release from the hypothalamus, while this corpus luteum is also making inhibin, and that inhibits the production of FSH. Now that both FSH and LH levels are low, that is going to, and so are the levels of estrogen and progesterone, that is going to, again, literally kill off the cells of the endometrium, leading to their shedding. Here is the relationship between what is going on in the ovaries, relating it to what is going on in the uterus. Okay, so the ovarian phases, pre-ovulation is the follicular, post-ovulation is your luteal phase. The follicular phase is made out of the menstrual part, so the shedding of the endometrium. And then there is the proliferative part. So this is under the effect of estrogen, where the uterus is proliferating. Okay, there is an increase in the size and the layers, the number of the layers of the cells of the endometrium. Okay? Hence the name proliferative, and again, it's under the effect of estrogen coming out of that follicle. Now that ovulation occurs, the corpus luteum is going to be making more progesterone than estrogen, and that increases the amount of blood vessels and the amount of glands that are there, so the mucus glands and mucus secretion. And that is why this part is known as the secretory phase. So again, your follicular phase in the ovary coincides with the proliferative phase and menstruation, while the luteal phase coincides with the secretory phase in the uterus. So this is the summary of the menstrual cycle. There, you can kind of go through this if you kind of read that. There's nothing new on here that we haven't talked about yet. So what are some of the effects of the female sex hormones? We'll talk a little bit about estrogen, and I believe the next one is on progesterone. So estrogen, and I don't want to read this whole thing, but I'm kind of focused on a couple of different things here. So estrogen is going to, as I said, it leads to the proliferation of the endometrial lining. It also leads to a watery cervical mucus. So mucus in the cervix can either be very watery, okay, and that is due to the effect of estrogen, or it can be very thick. Having a very thick, viscous or very thick mucus can prevent the sperm from reaching the egg. So in order for fertilization to occur, estrogen helps that process by making a watery cervical mucus. Okay, estrogen is also responsible for the external changes that occur during puberty. It's also responsible for that growth spurt that happens to bone, and then the ultimate stop of bone growth due to the closure of the epiphyseal plates. Okay, estrogen is protective against osteoporosis. That's why osteoporosis tends to get worse in postmenopausal women when there is no more estrogen or less estrogen. Estrogen also leads to vascular effects, especially to the blood vessels of the skin, and that's why, again, in menopause, there are hot flashes due to the lack of estrogen. Estrogen is also protective against atherosclerosis, and that's why atherosclerosis, again, or menopause, is a risk factor for atherosclerosis in women. Some of the effects of progesterone. Well, progesterone increases the secretions and blood vessels in the uterus, and that helps prepare the uterus for a possible implantation. Okay, progesterone makes the cervical mucus very thick and very sticky. And again, that is to protect, if fertilization did occur, that would be to protect what is now an implanted embryo. Okay. It will decrease the contractions of the fallopian tube and the myometrium. So again, kind of, protecting that possible pregnancy. It does stimulate breast growth. So during the luteal phase, during the phase where progesterone has the upper hand, there is sometimes breast tenderness due to the stimulation of breast growth. And it also increases body temperature. So one of the ways that some women, if they're trying to get pregnant, where they can find out if they ovulated or not, and again, ovulation means that progesterone levels are now high, would be an increase in body temperature. Not a fever, just a slight increase. So what happens during puberty in females? So the age range, again, it's very variable by ethnicity, by families, but the range is about 10 to 12 years of age. Menarche, that's the very first menstruation, okay, and that is one of the last events that happen in puberty. So puberty is kind of like a process that happens. It's not an overnight thing that occurs. There is the enlargement of the breast tissue. There is the presence of pubic hair and axillary hair and so on. So menarche, or the first menstruation to occur, is really one of the very last events of puberty. The average age of menarche in the US is about 12 and a half. Precocious puberty would be a very premature appearance of the secondary sexual characteristics, and that is usually due to an increase in the gonadal steroid production. So again, steroids are being made. All of these steroids can be converted into sex hormones, and that can lead to this precocious puberty. And that concludes chapter 17.